The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galleon Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, best digital health, best incubator accelerator, best startup.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galion Foundation.
Well, let's go right to the envelopes. I know dinner awaits. Don't worry, I'm experienced at this. I do want to say first that all innovations on display tonight deserve our respect. There are no losers here. Selections were made by the 50 members of the Prix Galien Awards Committee worldwide, distinguished scientists, all whose only incentive was to select the best. All these products have withstood the test of time for 50 years. Generations of patients do not lie. With that, this is more nerve-wracking than the Oscars. With that, I am pleased to announce our first gold medal for excellence in orphan and rare disease is Kimriah, Novartis. Next, for excellence in biotech, the gold is Herceptin, Roche. The gold medal for medicines, for pharmaceuticals, is Gleevec, Novartis. The gold for vaccines goes to Gardasil and Gardasil 9, Merck and C. And finally, in medical devices and technologies, the gold medal goes to Freestyle Libre, Abbott. Congratulations to you all. Now, may I ask the 5 of you to join me here on stage? I have to say, this really is more exciting than the Oscars. You know, it's one thing to make a movie, to keep people feeling their joy and joining us in our compassion and empathy. It's another thing to save people's lives. Very, very proud to be here and witness this. Let's give another hearty round of applause to your medal winners for the last 50 years in medicine.
Good afternoon and welcome, everyone. My name is Nafiz Zawahir, and I'm the Chief Medical Officer of Razorfish Health. It's a division of Publicis and Publicis Health. It's a great honor for me to serve as chair for today's second media presentation session for the Galleon Foundation, honoring the winners of the Prix Galien Golden Jubilee gold medals across 5 categories. In a sense, it's the best of the last 50 years. And I have to say, it's quite a way to start my year, to be in the virtual company of the leaders gathered here today, who are going to have an opportunity to share some of the highlights and challenges their organizations face in bringing these incredible innovations to market. When I think about the world we live in pharmaceuticals, biotechnology, medical device industries, I can think of no other industry, no analog where science, innovation, technology are harnessed for the betterment of the human condition. It is an awesome opportunity and a grave responsibility. So, which is why each year the Galleon Foundation convenes its global community to realize its mission to foster, recognize, and reward excellence in scientific innovation to improve the state of human health. And it does this through a variety of means. The first, and the reason we're here today, is recognition of outstanding private sector achievement in the discovery and development of new treatments for deadly diseases, chosen by an independent committee of 12 scientists, researchers, and academic leaders, including 3 Nobel laureates. Also by sponsoring biopharmaceutical, vaccines, med tech, and digital app awards programs in the US and across the globe in 14 other countries in the continent of Africa through the Prix Galien International Awards. But additionally, by hosting public forums to advocate for best practices supportive of greater private sector and biopharma innovation. The Galleon USA Forum, which many of you know, is held annually in New York, is just one example. And there, among other topics, we hear from the world's leading healthcare organizations and thinkers to discuss the past, present, and future of human health. And finally, by promoting partnership between the private sector and key public stakeholders like the government, academia, NGOs, patient groups, and the media. In this regard, the Foundation's Pro Bono Humanum Award, launched in 2007 by Galleon co-founder, author, and Nobel Peace Prize winner, Dr. Elie Wiesel, has made it a rising, influential force in the global movement for health equity, where health innovation is advanced as a human right, health for all. So now to the representatives of our 5 gold medal winning companies. As I'm sure you can imagine, the awards committee had an incredibly challenging task of looking back over 50 years of innovation to award the gold medals in 5 categories. And those categories are Best Biotechnology Product, Best Pharmaceutical Product, Best Medical Technology, Best Orphan or Rare Disease Product, and finally, Best Vaccine. So I'm very honored to say that we're going to hear now from the 5 representatives of those Golden Jubilee Gold Medal Award recipients. So I'm going to ask each of our representatives to talk to us about what it took to get the winning product over the finish line to commercial success, the clinical benefit of the product to patients, as well as the social and economic gains for society. So first, I'd like to introduce Dr. Gail Lewis of Roche Genentech, who will speak about the company's win for Best Biotechnology Product, the game-changing HER2-targeting monoclonal antibody, Herceptin.
Yes, thank you very much for that introduction. I'm Gail Lewis. I'm a senior principal scientist at Genentech in our Discovery Oncology department. And I am just very honored to be representing Genentech and Roche to receive this just incredible honor for Herceptin, or trastuzumab, as it's also known, for the best biotechnology product in the last 50 years. This award is very, very deeply meaningful to me as well, because I've been at Genentech for decades, and I was actually lucky enough to be a bench scientist at the time we started working on the HER2 antibodies, and was very directly involved in the preclinical development of Herceptin. So this just brings me great joy, I have to say. So this whole story started, actually it's interesting, the year I was hired at Genentech, 1985, was also the year that the HER2 gene was cloned at Genentech in Axel Ulrich's lab, and shortly thereafter was shown to be an oncogene. So it had oncogenic properties when tested in laboratory experiments. And this was really, and then following that, our collaborator at UCLA, Dennis Slayman, really gave us the seminal findings of showing that the HER2 gene was amplified at both the DNA level and overexpressed at the protein level in about 20% of human breast cancer, and that this amplification of the HER2 gene was correlated with a very poor outcome, with aggressive disease and a very poor prognosis in patients. Given that the HER2 is a receptor tyrosine kinase, it has an extracellular portion, made it very attractive to develop potential antibody-based therapies for this very potent oncogene in breast cancer. And so we set about at Genentech developing a panel of antibodies, one of which was then further characterized and became the antibody trastuzumab or Herceptin. Now, at the time, antibody therapy had been sort of tried and was full of failures in the field. And one reason for this is because the antibodies that were used were mouse, because monoclonal antibodies back then were all of mouse origin. So Paul Carter at Genentech actually developed the technology to humanize antibodies for use in patients. And this reduces the immunogenicity as well as allows the interaction of the antibody FC portion, the end that doesn't bind HER2, to bind and attract immune cells in your immune system to actually directly kill the cancer cells. So this was a real game changer. This was the first humanized antibody that was approved, and the first antibody approved for a solid tumor indication. And I think really ultimately laid the foundation for further development of targeted therapies. Now, in my mind, I think there are three really pivotal moments in the development of Herceptin. One was the experiments we did in the laboratory that showed that the activity of this antibody in inhibiting the growth of tumor cells was only demonstrated on cells that had amplified HER2, that had overexpressed HER2. This allowed the build-in of a companion diagnostic so that only patients that were considered what we call HER2 positive would receive this agent. And I think this is really key, because had this drug, Herceptin, been given to a broad patient population that were both HER2 positive and HER2 negative, it would have likely never been approved, because it would have been diluted out, as only 20% of breast cancer is actually HER2 positive. I think that was a pivotal sort of preclinical finding that led to the clinic. And then in the clinic, the first trial that was done that led to approval showed that in HER2 positive metastatic breast cancer, that the combination of Herceptin plus chemotherapy improved survival versus chemotherapy alone. But it was very important to follow that study up to go into early breast cancer, so in patients whose disease has not metastasized. And in the first adjuvant trial that was done with Herceptin, and again, it was shown that Herceptin improved median survival when combined with chemotherapy, compared to chemotherapy alone, and actually reduced the risk of the cancer coming back by about half. And these data were presented at the ASCO meeting in 2005. The audience gave this presentation a standing ovation. And the chair of the session, George Sledge, who's a breast medical oncologist, actually, I quote him, said, "Biology has spoken and we must listen." And I think that was a mantra for all of us developing targeted therapies, that this approach should work and we should definitely move forward. Now, that was just the start. Since then, a second HER2 antibody was developed that has sort of differing and yet complementary mechanisms of action to Herceptin, and is given in combination with Herceptin and chemotherapy in both the neoadjuvant, adjuvant, and first-line setting. And then as well, the antibody part of Herceptin, which is trastuzumab, was used to develop a HER2-directed antibody drug conjugate, wherein you conjugate a very potent chemotherapy type agent, you link it directly to the trastuzumab antibody with the hopes of delivering this potent chemotherapy agent directly to HER2 positive tumor cells while sparing normal tissue. This antibody drug conjugate is proved in both early and metastatic breast cancer, and as well has laid the trastuzumab part of the antibody drug conjugate, has again laid the foundation for the development of additional HER2-directed ADCs, one using trastuzumab, one of which has been approved, and there are several others that are in clinical development. And so I think the impact of trastuzumab, it has been significant. I really think the discovery of trastuzumab and its selectivity for HER2 positive breast cancer really laid the foundation for targeted therapy, really revolutionized the treatment of breast cancer. Because really until then, breast cancer was sort of considered one disease. Now that we can stratify these, we know we have the 3 subtypes of breast cancer, and HER2 positive breast cancer is one. And the identification of that actually helped us to identify these different subtypes of breast cancer. And again, before HER2 targeted therapy came about, the HER2 positive disease was a very aggressive disease with a very poor survival. And now with the great success of these HER2 targeted therapies, actually the prognosis for outcome of patients with HER2 positive disease is really better now than the other breast cancer subtypes. And just to give you an example, in the metastatic setting, the median survival in the first line setting has increased from 23 months with chemotherapy alone to 56 months. It's a huge difference with chemotherapy plus HER2 directed agents. And in the early stages of disease, if you give these agents in the adjuvant setting, about 9 out of 10 patients will have survival beyond 8 years. So I think the impact of all of this has been significant in terms of patient care, patient survival. And then I think one final thing I will say is that recently a subcutaneous form of Herceptin has been approved. So these drugs are usually given IV, and the advent of a subcutaneous form of an antibody allows for patients to go into the infusion clinic very quickly, come out very quickly. The treatment is fast. The treatment is non-invasive compared to intravenous injection. And most importantly, I think this had a real impact during COVID when it was very difficult for patients to spend hours and hours in the infusion clinic. So overall, I'm very, very proud of this drug, having worked on it from its inception. I think it's had a huge impact in the breast cancer community by extending survival with a non-toxic type of treatment, and has really laid the foundation for a number of HER2-directed therapies that have followed the approval of Herceptin. So I thank you again very much on behalf of Genentech and Roche for this incredible honor. Thank you so much.
Thank you, Dr. Lewis, and I'm glad that you note that trastuzumab continues to be the discovery that keeps on giving. I'm excited about the future as the achievements you've had in the past. Thank you for accepting the award, and thank you for that discussion. Next, it's Dr. Mark Taub, who's the Divisional VP for Technical Operations at Abbott. And he was one of the driving forces behind the development of the Freestyle Libre 14-day flash glucose monitoring system, which is the winner in the best medical technology category. Dr. Taub.
Yep, thank you so much for the introduction and for the opportunity to be here on behalf of everyone who helped develop the Freestyle Libre platform, portfolio products. Let me start by saying that I speak for all of Abbott when I say that we're humbled to receive this recognition, so thank you. The Freestyle Libre 14-day system is a significant health tech advancement for the people with diabetes. The system is accurate, it's affordable, it's accessible, and it continues to transform the way people with diabetes manage and achieve better health outcomes. Around the world, the Freestyle Libre portfolio has provided people with diabetes with accurate information they need around their glucose numbers, when they need it to make important health decisions without the burden of finger sticks. People who use the Freestyle Libre system are able to achieve more time in their target glucose ranges. They spend less time in dangerous levels of hypoglycemia and hyperglycemia. They see reductions in their A1C, a measure of their average glucose, over the last 90 days or so. They see reductions in hospitalizations and improvements of the quality of life, all without the burden of traditional finger sticks, which we know are painful and people don't do as often as they should and aren't discreet. While the development, detailed technology of the Freestyle Libre system took years to achieve, the system is now used to change the lives of more than 4.5 million people across more than 60 countries. So, thank you for the recognition of Freestyle Libre as the best medical technology of the last 50 years. So, incredibly grateful for the recognition. Thank you.
Thanks, Dr. Taub. Appreciate that. Next, I'd like to introduce Renaud Capdeville, who's the Global Program Head for AML and MDS at Novartis, who will speak to the drug that transformed leukemia treatment, our winner for Best Pharmaceutical Product, Gleevec, mesylate.
Thank you very much, and also thank you so much to the Prix Galien Foundation for this nomination that really recognize a true innovation in cancer therapy. So let me tell you a little bit about the story of Gleevec. So Gleevec is a tyrosine kinase inhibitor that is targeting several kinases, including what is called the BCR-ABL kinase. And this is very important because we are talking about chronic myeloid leukemia. So CML is a disease that was recognized in the 60s as being associated with a genetic aberration that was called the Philadelphia chromosome because it was discovered at that time in Philadelphia. And in the 80s, it was discovered that this genetic aberration was actually linked to the fusion of two genes, BCR and ABL, that led to an aberrant activation of the Abelson, the ABL kinase. And then that discovery triggered a question, would it be possible to inhibit that kinase or to target that kinase and develop a cancer therapy? In those days, I'm talking about the 80s, many people were very skeptical that that would be achievable to have the right level of specificity to deliver a meaningful therapeutics. However, that's where the story begins at Novartis, or I would say at the preceding founding company of Novartis, Ciba Pharmaceuticals, a company that started in the late 80s, a research program on tyrosine kinase inhibitors led by Dr. Alex Matter. And one of the first targets they worked on was actually BCR-ABL. And that led to the discovery of Gleevec that at the end got approved by the FDA in 2001. So what does it mean? And why Gleevec really changed the way we think about cancer therapy? So in CML, before Gleevec, the patient with this disease usually survived between 3 and 4 years. So that was quite a short survival, and the treatment was based on interferon, a drug that was very difficult to give with a lot of side effects. Now, after Gleevec, the life of this patient became quite close to normal, with a very good safety profile, allowing a lifelong therapy. So a radical shift from a super short survival to a near normal survival. And even more than that, some patients responded so well to therapy that it became possible for some, unfortunately not all patients, but for some patients to discontinue treatment and observe what we call treatment-free remission. And that was very important, particularly for young patients, like young women who wanted to become pregnant. So really a drug that completely changed the way to think about the management of leukemia and was the very first treatment to validate the concept of targeted therapy of cancer when there is a genetic aberration that is actionable therapeutically. And that led the way to the introduction of other targeted therapies later on, like in skin cancer and lung cancer and other disease. So that's a little bit what is the story of Gleevec. And today we have a second and third generation ABL inhibitors that have completely revolutionized the treatment of CML. So again, a real privilege to talk about Gleevec today. That is also as a personal meaning to me because I had the privilege to be the clinician in charge of the clinical team who delivered the initial clinical studies in CML and other indications. So thank you very much.
Thank you, Dr. Capdevielle. I still think that there was a stunning statistic that I remember learning as a younger man. That someone with CML who's in remission after 2 years of Gleevec treatment has the same life expectancy as someone who doesn't have cancer, which is just a mind-blowing sort of statistic to think about. So thank you for that. Let's move on to our next panelist, Dr. Abbey Bookbinder, Global Program Head of Novartis, and he's going to speak to us about the company's win in the Best Orphan or Rare Disease Product category for the first FDA-approved gene therapy, the CAR-T, Kimriah.
Thank you. So yes, I'm Abby Bookbinder, a global program head at Novartis, and I am so proud and fortunate to be able to describe the product Kimriah, which in fact is a cellular therapy that is modified through gene therapy. And thank you so much also to the Prix Galien organization for recognizing the benefits that Kimriah brings to patients. So, what did it take in order for a product that was an academic product to really become a commercial success? So, historically, what happened is there were data that were generated at the University of Pennsylvania, a very big academic center in the United States. What Novartis did was really to follow the data that were generated at that institution, first in patients who were children with acute lymphoblastic leukemia, and then very shortly thereafter, in adult patients who happened to have lymphoma. The approach that we're talking about was really revolutionary. It was a very big risk to take a look at what was going on with 3 patients in a published report, and then make the decision that there's going to be investment, and this is going to be something that is really going to be transformational to patients. And this approach really reprograms the cellular component of our immune system in order to be able to specifically target cancers that express a specific target on the surface of their cells. This reprogramming is in fact very complex, and it's part of what it takes in order to bring this to a commercial success. The complexity is that cells have to be taken from the patient, T cells, part of the cellular immune system, and then outside of the patient have to be manufactured in order to reprogram these cells to then be able to target the cancer, bring those T cells in close proximity to the cancer, and have those T cells kill the cancer with the target on it. So what did it take? I think that it took courage, it took a lot of perseverance, and really it took a lot of maintaining the course through many ups, many downs, and many challenges, including clinical challenges, including manufacturing challenges, including continuing to have a belief that what was seen at the beginning is going to translate to what we are going to have at the end. It took continued investment. It took continuous improvement in manufacture to be able to bring a product that was manufactured in very complex way to very few patients, but now this product has to be able to be manufactured to patients on demand for an approved clinical product after the completion of the clinical trial. And this is really what it took. Now, at Novartis, we're also continuing to work on these kinds of CAR-T therapies to bring products that may have even a greater potential for long-lasting remissions from lymphoma and leukemia. What is the benefit to the patient? So this was truly revolutionary. This has the potential for a cure, right? Not for every patient, unfortunately, but for many children who had no hopes, no other therapies that could be given to them. All the previous therapies had failed. It's the potential for a full life, a full long life, and a full productive life as a member of society with a one-time therapy. The therapy gets given, there may be side effects, and then there is the potential for this long productive life, which is an incredible benefit to the patient, to their family, as well as to society.
Thanks, Dr. Bookbinder. If anyone has the chance to read the narrative of the first patient to receive a CAR-T therapy, it reads like a movie. It looks like it's like a movie script. It's a truly beautiful story, and just a wonderful testament to what we can do, when industry and academia works together. Finally, last but certainly not least, we're going to hear from Dr. Richard Haupt, who's the Vice President, Head of Infectious Disease and Vaccines, Global Medical and Scientific Affairs at Merck and Company, here to talk to us about the best vaccine of the last 50 years. And I think it's to no one's surprise that it is Gardasil, and Gardasil 9, of course, which has dramatically changed the incidence of cervical and other HPV-related cancers worldwide. So excited to have you here, Dr. Haupt. Please go ahead.
Yeah, thank you very much. And, I've spent about 20 years of my career at Merck supporting Gardasil, either in the medical affairs or clinical research side. And I'm really honored to be here today on behalf of Merck, and really want to thank the Galion Foundation for this recommendation. I think we all know that effective vaccines can have such an important impact on public health and society. And so to be recognized with this Golden Jubilee Award for Gardasil and Gardasil 9 is really a proud moment for Merck. So let me first talk about what it actually took to get Gardasil over the finish line, as it were, for commercial success. And I think it's notable, it was decades in the making. The first piece of this was the recognition that HPV was a cause of cervical cancer that was proposed by Harold Zerhausen in the 70s, but took another decade before it was validated, and he ultimately won the Nobel Prize for that. But it took many years of work by scientists around the world and Merck to make an effective preventive vaccine a reality. We began our first phase 1 trials back in 1997, and Gardasil was eventually approved for use in females in 2006 by the US FDA, and then approved globally in many, many countries around the world. And that first vaccine targeted 4 HPV types, the 2 most important types associated with cervical cancer and 2 types associated with genital warts. And the reality was there was certainly no guarantee when we started developing this vaccine that it would work. Obviously, not only did it work, but the data was really remarkably strong. So we had a vaccine that would protect against cancer, some cancer-causing viruses, and we knew this could have a tremendous impact on public health. And just as a reminder, Gardasil consists of non-infectious recombinant virus-like particles of the L1 protein of HPV. It's a very complex vaccine to manufacture. And so part of our investments were heavily involved in the technology and the manufacturing capacity to be able to have enough vaccine to support the world. And we haven't stopped there. Recognizing the potential impact on public health, we've continued to investigate other potential benefits of Gardasil, and we filed for use, and we have it approved now for boys and men. And we've identified additional cancer-causing HPV types and wanted to add those. And so in 2014, Gardasil 9 was approved, and that covers 5 additional oncogenic HPV types. And then most recently, we've received an accelerated approval in the US for the use of our vaccine in the prevention of certain HPV-related head and neck cancers. And we have a large confirmatory clinical trial underway to support that accelerated approval. So bringing both Gardasil and Gardasil 9 across the finish line was a complete team effort that involved many, many individuals in our organization, but certainly was very much based on the discoveries of those who came before us. So let me turn briefly to the clinical benefit, as well as the social and economic gains. We've distributed hundreds of millions of doses around the world since Gardasil's introduction in 2006. It's really hard to measure the impact, but we're now starting to see more and more what this vaccine can do from a public health perspective. And we recognize that every cancer case this vaccine prevents is a mom, a dad, a brother, a sister, a son, a daughter to someone. And so it's really, really important. Our clinical trials demonstrated efficacy of 98% against cervical cancer, 100% against vulvar and vaginal cancers. We also demonstrated efficacy against anal cancers in males. And as I noted, we are now studying head and neck cancer as well. And we've begun to see in the last 5 years or so some really startling real world vaccine benefits. We've recently, in fact, just published our own literature review of close to 100 real world studies that have shown Gardasil resulting reductions in vaccine type HPV infections, as well as HPV related diseases, including cancer, precancers. We've also seen real world data from others. Just recently, the American Cancer Society showed that between 2012 and 2019, there was a 65% reduction in cervical cancer incidence rates in women in the United States between the ages of 20 and 24. These are some of the first women that would have been eligible to receive HPV vaccination. In 2019, Australia showed a 50% reduction in cervical cancer in women aged 20 to 24 years of age. And that followed also a large-scale vaccination program of Australian girls. We continue to collect data and study all of this, but certainly the findings today indicate this vaccine is having and will continue to have a very huge impact around the world. our work is not done. We continue to recognize that much of the global population who is eligible for HPV vaccination don't receive it. And this is particularly true in lower and middle income countries. So we're really working very hard to increase our manufacturing capacity. We have agreements with Gavi and UNICEF to provide over 90 million doses to access these countries through 2025. And again, we are so hopeful that we can continue to deliver Gardasil and Gardasil 9 to have a huge impact on populations and societies around the world. Thanks again for this really remarkable honor for a truly remarkable vaccine.
Thank you, Dr. Haupt, and thank you to all our panelists today. I want to open this up right now. We have an audience who might have questions. I want to invite our panelists to ask each other questions. And maybe I'll kick it off a little bit. We discussed right before the panel started that one of the most exciting things about all of these innovations are not just the achievements that they have already made, but the promise of the future. Maybe I'll start with you, Dr. Lewis. What makes you most excited about the future of trastuzumab?
Yeah, thanks for the question. I think what I'm most excited about is the way that innovation continues to happen in the HER2 space. So, trastuzumab was first approved in 1998, so it's been a few years since then. But people are developing new modalities. Like I mentioned, there's antibody drug conjugates. There are immune cell bispecific antibodies. There are small molecule kinase inhibitors that target the intracellular kinase domain of HER2. There's combinations thereof of all of these modalities, and including combinations with immunotherapy agents. And so I think there's a lot, we have done very well in the HER2 space in terms of treating patients, but there's room for improvement, certainly room for improvement in the metastatic setting, room for improvement in the neoadjuvant setting to get better pathological complete response rates. So I think it's up to all of us as a community to continue to discover these new modalities and actually really do the right studies to determine combinations and combination partners for the modalities that we have, so we can really, really improve further the outcome of patients that have HER2 positive cancer.
Wonderful, thank you. I'll ask the same question to you, Mr. Buchbinder. CAR T is a huge advancement. What are you looking forward to as your next big step, the next big achievement for cellular therapeutics?
So yeah, there are multiple areas where there is the need and the potential for CAR T, where there's a medical need, where there is a potential for CAR T cells to be able to improve. So just taking a look at various targets, now that there is the proof of concept in the sense that one can take the immune system, reprogram it in order to target cancer for a specific target. In the case of Kimriah, it was CD19. We and many other companies are also working on saying, well, maybe this approach can also be used in order to target other targets that may be relevant to cancers that are not only lymphomas and leukemia. Early stage in the game, so we're not there yet, but I think that this is really important, and the demonstration that really can help patients with a curative potential is what is the aim when we take a look at other targets. There's a second thing, which is that when you take a look at the results of Kimriah, you may have noticed that I said there's a potential for cure. And the sad reality is that, yes, there are patients who I think are going to truly be cured. There are cases that we hear about in the news with the first patient, the second patient, et cetera, and many patients who now are going towards more than 10 years and towards 15 years where they receive the treatment one time and that's it. The cancer has never come back, which is almost a miracle. The reality is that there are some patients who receive this treatment and the treatment does not work, or it may work for a temporary period of time and then it comes back. So I think that even for CD19 positive cancers, that's the target that we are targeting, there's room for improvement. And we're also working on that by finding maybe some cells that may be more persistent, by finding ways of manufacturing the cell in order to really have those cells remain longer, be more active against the cancer. And I think that one of the realities is that this is right now a bespoke therapy. There is a patient that first gets identified, and then you manufacture the cells for that patient. That is very complex and it takes time. And do you have the ability to do this for a limited number of patients with more rare cancers such as leukemia and lymphoma. What happens if this is for lung cancer, for breast cancer, for colon cancer, where you need to be able to do this for a lot of patients? So I think there's a manufacturing science that really needs to be done for those bespoke therapies. And then I think that finally, there are many, many companies that are also working on this concept, but by saying, look, the concept is there is a T cell, and the T cell gets reprogrammed to attack specifically to target the cancer. Can we take just a general T cell of a healthy person and find a way to make this cell target the cancer? And then we find a way that this T cell is accepted by patients, does not attack the patient, is not rejected by the patient. So there's a lot that really now is able to move forward now that this first step, this proof of concept, if you wish, has taken place. And I think we're living in a very exciting moment, and let's see where we're going to land.
Fantastic. So it sounds like the future is both, to borrow a fashion term, but more bespoke and more off the shelf. And that's an exciting place to be in cellular therapeutics. You know, Dr. Capdevielle, so CML, what's next? What's next for you and for Gleevec? Where are you going to go? Where are you going to go next?
Okay, no. So, I mean, obviously, you could say that after the success of Gleevec, many people thought, okay, now CML is done. Let's move to something else. We have bigger challenges. But in reality, that was not the case. And actually, I think it was a privilege to be in Novartis over the last nearly 30 years, because after Gleevec, the company continued to invest in CML research. And that led us to a second generation TKI with Tasigna or nilotinib that was more specific to the target, and with this had a better efficacy than Gleevec. But that was also not enough, because we started to understand that there are some mechanism of resistance that comes in, and some patients actually don't do too well, even with these great therapies. And that led the Novartis researchers to develop another drug addressing another aspect of the kinase. And the drug has been approved a couple of years ago by the FDA and the European agencies and other regulatory authorities. And the drug is asciminib. So we are already in the next wave of innovation in CML. But now I think researchers in that field are asking a very important question, because, as I said earlier, a minority of patients can achieve what we call the treatment-free remission, which means that they can stop treatment, and then some of them might be cured of their disease. It's too early to say, but that's a possibility. But it's only a minority of patients. How can we make many more patients to go there? And it's a very complex challenge. Perhaps that will require other approaches, like some kind of immunotherapies. It will also require a better understanding on how best to monitor patients. Today, we still have a challenge in that area, that many patients with CML don't have access to PCR testing that is so critical to make a decision for one or another therapy. Even in a country like the US, there are still too many patients who don't have access to these tests. So that's a challenge that has to be addressed at the global level, because that really could improve the care of this patient and allow more patients to be candidates for stopping treatment. Amazing. So in other words, the next challenge is how to stop treatment, which is a good challenge to have.
Which is a good problem to have. Thank you, Dr. Capdevielle. Dr. Taub, you know, when I think about the Freestyle portfolio, you know, it's across professional and patient. It's led to more freedom, better glycemic control, better health outcomes for millions of patients with diabetes, all while, you know, as you like to say, eliminating the choice between what hurts and what's healthy. You've achieved so much with the portfolio. What do you think the next horizon is for you and the Freestyle Libre?
Thank you. That's a great introduction, so I appreciate that. Diabetes is such a burdensome disease for people with diabetes. They have to constantly think about their glucose levels and what they're eating and if they're exercising or not exercising enough, what their medications need to be, if they're titrating their own doses of insulin, or if they're just remembering to take their medication. It's a huge cognitive burden. So Freestyle Libre platform, it provides patients with access to their glucose numbers, what their value is now, how they got there, their past, their history of glucose information, how it's evolved over the last 8 or 12 hours. It tells you where glucose is heading, but there's opportunities to link that glucose data to other data sources where appropriate, to things about meals and exercise, and provide even better insights to patients to better manage their diabetes and help reduce some of that cognitive burden. There's opportunities to help physicians and the patient care team for those patients who are difficult to manage with therapy insights that you can only begin to do once you have dense glucose data that you can use and really understand. It's not just when a patient comes in and what their logbook says or happens to say, and do they only test when they're good, or do they make up their numbers? This is the truth, and it's easy to get, and that information comes in, and patients make better decisions with it, and physicians make better decisions with it. But how can we make that even easier? I'd also say that when we developed the Freestyle Libre system, we knew we were really developing a platform. And while we use it right now to provide glucose values, to measure glucose, that there's an opportunity to look at other analytes as well, that there's lots of other things where people could benefit from continuous measurement of analytes of interest. And those are areas that we're exploring and discussing. And then the last thing I touch on is just that, the Freestyle Libre system is really designed from the start with access and affordability in mind. And that's not something that's always the case in the space. And we're incredibly proud of that. And it was why the Freestyle Libre system needed to be 14 days, and it needed to be no calibrations. That way, we could reduce the cost of the system and get it to a price where it could be widely reimbursed and get it in as many hands as possible. And I think that, we're beginning to see a change where it's not just, you develop the best product and then you charge more for it, but that you develop the best product and that you try to get it in the hands of as many people as possible. And we're incredibly proud of that, and think that that's an opportunity that we've seen with Freestyle Libre, and that there's even more to be had there. So yeah, thank you.
That's an awesome answer. Thank you. I would be remiss if I didn't point out too that the way you've designed the system really decreases the environmental impact of the product as well. So, when we look at, when I look at what you've done and what you've created, it's like proof of concept for how do we continuously monitor a chronic condition, whether or not an empowered patient makes better healthcare decisions, how all of these things go back to better outcomes for all. So again, congratulations. I think it's a wonderful achievement, and I look forward to the future as well.
Well, thank you so much. And we continue to advance it. So I think you're pointing out a Freestyle Libre 3, which is even smaller. It's the size of,
right.
2 pennies, 2 stacked pennies in the back of your arm, and we reduce the plastic waste and the paper waste, and better for the patient, better for the planet. So yeah, terrific. So thank you so much.
So much of what you have achieved, right? It reaches comparative to many of the other conversations around vaccine, you've seen incredible advancements in the uptake of Gardasil. The remaining ground we have to cover. What is the communication, the message we have to get out to people? Do you think the most important message around a vaccine preventable cancer that is, that people need to understand in order to maximize coverage with a vaccine like Gardasil?
I think that's a very complicated question, although it seems pretty straightforward. And the reason I say that, it probably varies depending on where you go and why. What the barriers for implementation are, because they're not all exactly the same depending on what part of the world you're in. And, frankly, if you look at the industrialized world, like the U.S. and parts of parts of Europe, there's still concerns about safety with the vaccine. There's a lot of misinformation around vaccine safety. Many of us are aware of this through much of the anti-vaccine work that has complicated our lives with COVID over the last several years. But that certainly exists for all vaccines for the most part. And there are some unique anti-vaccine messaging that occurs with Gardasil that has to be strongly combated and defended against. But if you look at the lower and middle income countries, frankly, that's where most of the unmet medical burden currently exists. It's really much more of an infrastructure problem, whether it's the cost, not really so much the cost of the vaccine as much as it is the cost of establishing an infrastructure to deliver the vaccine. And part of that is that the vaccine is delivered really predominantly to the adolescent population. And in many lower and developing world countries, they don't have an infrastructure to access adolescents. There are programs called EPI, expanded programs for immunization, that are really addressed at the infant age group. And so there have been great strides in vaccinating infants, but not so much in the adolescent population. And some of that is really about developing infrastructure, but a lot of it's the political will. And so a lot of the work that's required in many of those parts of the world is developing advocacy and strong support. And honestly, all the great science that's being generated to show how good Gardasil is, and that it can actually prevent cancer and we can move to a world that we can eliminate cervical cancer. So a lot of that is going to help drive that. So it varies quite a bit, honestly, depending on where you go. I would, when I was thinking about your original question, I was thinking, what's the most important? There are many avenues that Merck is working on, or we're supporting and helping, but I would say that trying to expand greater implementation access is still the #1 most important thing. I mentioned new indications, new populations, new data, but if we did none of those, but could get the vaccine into larger populations of adolescents, it would make a huge public health impact.
Thank you. I understand it's a complicated area and a complicated answer, but at the end of the day, we are so close to eradicating cervical cancer. And I know that if it's within our grasp, I know that we can do it. And I'm sure that Merck will get us there. As we come to the end of our time together, I did want to note a couple of things. Listening to everyone speak today, beyond just being amazed and honored to be here, I noted that seems to be a theme that runs through all of these innovations, right? And if I might be cute for a second, it really comes down to partnership, passion, and pragmatism. I think that I'll start with, I see all of you. I see the organizations that you come from. We're passionately committed to what you were trying to do, even in the face of obstacles, even in the face of difficulties. And I think that's really, that kind of commitment comes through. That idea of partnership, whether it's with patients or it's with physicians. I think about all of these products and the amount of close interaction, whether it's Carl June and University of Pennsylvania with cellular therapeutics, or it's Dr. Slayman with Herceptin. There's just such a wonderful partnership, the best kinds of partnership between academia and the private sector to, again, drive health innovation forward. And then, pragmatism. I think that, earlier on when we were talking, Dr. Lewis, you mentioned that part of what went right, that pivotal moment, was recognizing that if you try to, demonstrate the efficacy of this drug in an all-comers population, it's not going to work. You need to focus, and you need to be pragmatic about who you're going after. And that was one of the pivotal things of focusing on HER2. So again, passion, pragmatism, and partnership seem to come through with everybody. I want to thank everyone, congratulate all the winners on your incredible innovations. I want to thank all of our guests in the audience who joined us today for both of the webinars, this one and the previous. And as we begin 2023, we greatly anticipate seeing the novel, outstanding contributions that will be made in these industries and the continued success of the products we talked about today. Today's webcast will be available shortly for on-demand viewing on the Galleon Foundation YouTube page. And I'd like to thank everyone again, and congratulations to all the Golden Jubilee winners and to the Galleon Foundation for hosting today's webcast. Thank you all.
Thank you so much.